Thank you for joining this webinar discussing choice of endpoints on COVID trials. My name is Lisa Hickey, and I'll be moderating this webinar. Dr. Ken Ghosh will be our presenter. He's an experienced industry statistician with over 25 years supporting clinical trials across all phases of development. He's worked in both large pharma companies like Merck and BMS, as well as on the CRO side, founding both Physician and now Endpoints Inc. For this webinar, the first 45 minutes will be devoted to Dr. Ghosh's presentation. If you have any questions, please submit them using either the chat function on the right of your screen or email, and my email is on the chat. The remainder of the webinar, the last 15 minutes, will be used to answer questions. If you have additional questions or we don't get to your question, please feel free to follow up via email. So Kalyan, take it away.
Thank you, Lisa. And let me share my screen. Okay. I'm guessing you can see my screen. Give me 1 second here. Yes. Okay. So choice of endpoints in COVID-19 trials. Right now, there are over 2,000 clinical trials that are ongoing worldwide. They are sponsored by industry, governments, and academia. But out of those, about 200 or so are industry-sponsored trials. So we'll talk about, some of these trials, what the regulations are around the trials, and what is to look at as efficacy endpoints in these trials. Now, obviously, the middle box is the topic for today, but I'll also talk about the bottom box that you see there. So I'll set up the general overview of endpoints, how they're selected, how they're placed, and what does an efficacy endpoint mean first. Then talk about the middle box, and then go to the top box where I say other considerations, just to make sure that we put the endpoint in its proper context. Now, just to make sure that we are talking about more confirmatory studies here, late stage studies. We are not talking about early phase studies, phase 1 or phase 2, where the objective of the studies are quite different, choosing the dose, looking at interaction, etc., etc. Now, this is the current law around efficacy. This is United States law that the FDA follows. It's a double negative the way it's written here. So basically, it says that we have to have. I hope everybody can hear me. Now, the substantial evidence on something that we need. Now, what substantial evidence of what? So basically, it says if you don't have substantial evidence, then the drug would not be approved. And that substantial evidence is something clinically meaningful. Okay, so let's look at what does that mean. So clinically meaningful something is basically something that the patient can relate to something directly. And they fall into 3 categories. One is feel, the second is function, and then survival. Any efficacy endpoint has to fit into these 3 boxes. Okay, either the patient has to feel something, or has to function better, or obviously we know what survival means. Some of the examples for feeling, for example, an obvious feeling is pain. So reduction in pain would be an efficacy endpoint. For function, it could be that better mobility for the patient, and that would be an important thing to the patient, right? And then the ultimate clinical outcome is survival. Now, it is possible that we would combine, sometimes efficacy endpoints would combine some of these elements. So for example, in a like rheumatoid arthritis trial, the primary endpoint would be a combination of feel and function. Okay, so that's sometimes is done, which we call a composite endpoint. And also to remember that some of these endpoints could be subjective. We could ask the patient, subjectively how they feel on a scale, or sometimes they are objective, and obviously survival being the most objective endpoint. Now, so those are the three. Now, one of the things I have to remember is that those three endpoints, the patient may have them now, or they are going to be impacted by these three very soon. So if somebody has an infection, that needs to be treated, because otherwise it would lead to something where they will be compromised with respect to their feeling, function, or survival. There is an exception to these three, and that exception is called a surrogate endpoint. What is a surrogate endpoint? As you can see, the first two bullets, a surrogate endpoint is a lab measure or a physical sign that is basically a predictor of clinical outcome and can work as a substitute for a clinical outcome in a clinical study, in a confirmatory clinical study. And there are some examples, for surrogate endpoints that have been widely used. For example, for HIV, CD4 count or viral RNA was used to get approvals for a lot of drugs. Now, the one issue with surrogate endpoint is that for an endpoint, so the endpoint could be a marker of some kind, right? And for that to be used as a surrogate, it would require strong data to validate that the surrogacy works. So which basically means that if you have improvement on the surrogate endpoint, then you will have improvement on the clinical endpoint. And that has to be proven in a study. And this is not an easy task, of course, and there are various literature on how to validate a surrogate endpoint. Again, obviously, you will need data within one trial where you collect both. You collect the clinical endpoint as well as the surrogate or potentially surrogate endpoint, and then show that one leads to the other. Now, usually, these surrogate endpoints sit on the therapeutic pathway. So we know that, for example, viral RNA for HIV is a good surrogate endpoint, because if we know that if we control the virus, if we lower its level, then it would impact the disease state, which is AIDS for HIV virus. And this is not so easy for all diseases. I mean, for HIV, it has been proven that viral RNA is a surrogate endpoint, but this is not so in COVID-19. So this is not a given that anytime you control the virus, you control the disease. And I'll talk about that a bit more later. Now, these are some of the examples. On the, right now, there are actually 70 disease areas for where a surrogate endpoint is acceptable to the FDA. And I have pointed to 4 of these here. Now, for the, one of my first trials were HIV, and when the protease inhibitors were coming out starting in 1995, I think that was the first protease inhibitor. And at that time, viral load and CD4 count, they were used to approve drugs. And obviously, we know what difference it has made to the community. And obviously, the natural question would be for COVID-19, is that there a surrogate endpoint on, or what to do about it, or what is the regulatory pathway for that? So in the next slide, I talk about the pathway. So the first bullet, you can see that the surrogate endpoint can be used for drug approval. So this is part of the law. If the surrogate endpoint is well validated. This is what we just talked about, and this is a complicated process by itself. But then the interesting thing is it says or. So obviously the endpoint doesn't have to be well validated. There is another opportunity here to use an endpoint as quote unquote surrogate for the time being. And you don't have to read the whole thing, it's too many words there. But what it basically says that even if it is not well validated, an endpoint can be used as a surrogate if it is reasonably likely based on therapeutic physiologic evidence that it can predict clinical benefit. So it's not quite validated, but it's reasonably likely from the way the drug works. But there is one catch, that is that you can get approval, but you have to make a commitment to the FDA. And I'm talking more about FDA in this talk because other regulatory agencies are pretty similar, that you'd have to show, basically commit that you'll do another trial later on to make sure you prove the surrogacy. Okay. Now, let me so this is the setting for a surrogate endpoint. So in the COVID trial, we might be able to use something that is reasonably likely. Now, what is reasonably likely is not clear. That depends on the mechanism of the drug, how the drug works. Because as I said, usually reasonably likely means that it has to sit on the pathway how the drug is supposed to work. And this might need some discussion with the agency if a sponsor wants to use an endpoint that they think is reasonably likely, whether the agency would also feel that they are reasonably likely. Now, types of endpoint. So we talked about the definition of endpoints, right? We talked about field, function, survival, and then surrogate endpoint. So that's one way to look at them. The other way, especially for us statisticians, we look at endpoints as whether it's quantitative, it's numerical, or it's qualitative, because that's how the design of the study, how big the study should be, how you'd analyze the data, etc., would depend on. For a numerical endpoint, obviously it could be continuous or discrete. Some of the examples would be visual analog scale. Some of the discrete ones could be some kind of scoring. It can be patient-reported outcome as well, quality of life, SF-36 or something like that, right? So these are some of the examples. And then the other numerical endpoint is time to event, time to death, time to discharge from hospital. For example, in COVID, these would be endpoints of interest that we could look at. So here, the number is basically time, number of days, number of weeks, number of years, months, etc. And for qualitative endpoint, the simplest qualitative endpoint is a binary endpoint. That's a success or failure, death, or being alive. Or sometimes it could be ordinal, which is like degree of impairment, for example. It's like, mild impairment, moderate impairment, severe impairment, etc., where we know that one is worse than the other, but they're not really numbers. Okay, so for example, sometimes in COVID trials, I'll talk more about ordinal endpoints there. It could be, we know that death is worse than ventilation, ventilation is worse than, hospitalization, hospitalization is worse than no hospitalization. So there is a natural order there as well. Now, when we have these three, so I have three boxes here. We talked about what is being measured: survival, filling function, surrogacy, etc., surrogate endpoint, etc. We talked about what type of endpoint. It could be continuous, it could be categorical, ordinal, time to event, etc. And then I will talk about how do you place the endpoint? How important is it? Is it a primary endpoint? Is it a secondary endpoint? Is it an exploratory endpoint? And there are some regulations around that, how you would place an endpoint in which box. So I'll come to that in the next slide. But I think the point here is that you can choose an endpoint. Let's say we choose survival, right? Then there are two choices. Do we look at survival as a categorical, as a binary thing, like, proportion of people dead, a proportion of people alive, or we look at when do they die, time to event, right? Do they survive for 5 months? Do they survive for 5 years, etc.? And we can put survival as the primary endpoint either way. And that choice we'd have to make depending on what the disease is and what is clinically important. Or if we say feel, for example, could be a continuous endpoint, and we can put it as a secondary endpoint. Now, what are the primary, secondary, and exploratory endpoints? So there are these are the three baskets, right? So the primary endpoint, the success of the trial would depend on it. So if you fail on primary endpoint, your trial has failed. Obviously, you'd learn things from the trial, but overall, as a confirmatory trial, that trial has failed. And we, to make sure that it doesn't fail, we power the study mainly for this endpoint. And we can power the study for secondary endpoints as well, but the primary endpoint is a must. Multiple primary endpoints can be used. We call them co-primary endpoints if both of them must be met. And if we design a trial where we look at two different things, let's say hospitalization and mortality, or two different things, and if we want to say that we will prove at least one, then you will need what we call a multiplicity adjustment. Okay, because when you get two chances to be successful, there is a penalty that you'd have to pay through that, what we call the type 1 error rate, or the adjusting the false positive rate. For secondary endpoints, if you want to include something in a labeling claim, it must be included in either the primary or the secondary endpoint as an endpoint, right? So again, the multiplicity concerns are also applicable to secondary endpoints, right? You have to pay attention to what we call family-wise error rate, or you can loosely say false positive rate. And in COVID trials, I see some of the trials that are already in the public domain. Sometimes you see 20, 25 different endpoints which are called secondary endpoint. I mean, they're okay, but without any multiplicity adjustment, it would be difficult to place them in the product label. And the other bucket, the exploratory endpoint, can be anything. You don't need multiplicity adjustment. These are for learning and for preparing for the future. So that's the basic, overall general overview of how endpoints work. I did forget to mention one thing, that at the right hand top corner, you see, I have these three things because I broke the talk into three parts, and I want to make sure that you know where we are. Okay, so we were just at endpoints in clinical trials, right? Now, next, I'll get into the COVID-19 endpoints. So let's talk about COVID-19 a little bit. So COVID-19, the name comes from, probably most of you know, from coronavirus disease 2019. The co is from the coronavirus, right? The corona, V is for virus, and D for disease, right? And the name was chosen on February 11th by World Health Organization. And the name, there was some controversy about the name first, because first they were thinking about calling it SARS, but at that point, who thought that that would scare people, especially in Asia, because there have been so many deaths from SARS there before. So they selected the name COVID-19, but now we know that the death rate is, and the number of deaths is much more. So next time, probably it would be, if there is another coronavirus disease, it would probably have a different name. Now, coronavirus is not unique for COVID, right? There are different strains, different virus. I don't want to get into that, but the coronavirus is a generic thing, right, in some ways, right? And it can lead to different diseases. The COVID-19 disease can range from mild to severe, right? And for severe disease, it could lead to pneumonia, acute respiratory syndromes, and multi-organ failure, and ultimately death. The incubation period is about 2 weeks. The median time for symptom onset from exposure is about 4 to 5 days. What are the therapies for COVID-19 right now? Actually, there are no drugs or therapeutics presently approved by anyone, right? So there are basically clinical management with treating symptoms and some supportive care. These are some of the most drugs that have been talked about. And then the other clinical management thing is ventilation. Now, for ventilation, I'll just go quickly over this. So there is this mild ventilation, non-invasive ventilation on the left, which is basically a CPAP machine. And then you have the middle one where oxygen is pumped into the lung, and then carbon dioxide is taken out. This is mechanical ventilation. And sometimes people use mechanical ventilation as an important endpoint. Like time to mechanical ventilation, or what proportion of people go on to mechanical ventilation, etc. The last one, ECMO, that's a much more invasive procedure. Here, basically, the blood, is purified outside the body, even, and then circulated back into the body. So, which is, you can see, would be useful for extremely critical patients. Currently, this is from clinicaltrials.gov. I said about 200 trials are ongoing. This says, how many trials are where. You can see the US and Europe, most of the trials are happening there. Now, for the regulatory guidance for COVID-19 trials, there is an international coalition of regulatory authorities. They had a workshop and it came out with some recommendations. And FDA has published 2 guideline documents, one for vaccines, the first one. Obviously, vaccines are to prevent COVID-19, right? So it's for prevention. And then drugs and biologics that are for treatment or prevention, both, right? So the vaccine guideline actually is pretty thorough. It has 20 some pages and it goes from manufacturing to everything, right? It covers much more, whereas the drugs and biologics guideline is not, doesn't have as much material. It focuses only on the clinical side of things. Now, the FDA guidance, I think, covers pretty much everything that others have talked about. So I'm focusing on FDA guidance. That's why. So for example, if we go back one slide, the ICMRA, it's an international coalition. This workshop was chaired by PMDA and EMA. And if you look at the recommendations, they are not different from what is there in the FDA guidance documents. Now, for vaccines, let's talk about vaccines in a couple of slides. For vaccines, right now, the guideline is that the primary endpoint could be either the disease itself, which is COVID-19, or the virus infection. So either one could be acceptable. If it is COVID-19, then it has to be confirmed by virology. But if it is, so which basically means the presence of virus, that is measured directly through a PCR assay or something like that. Whereas the infection could be either virologically or serologically confirmed. So it could be either detected through antigens or antibodies. And sponsors also are recommended to collect the clinical outcome data, including hospitalization, symptoms, etc. And it is also important for vaccines if it can be used as a prophylaxis, uh, uh, you know, to see whether COVID-19 is milder in people who are receiving the vaccine compared to the ones who are not. Now, the other slide is, again, I mean, this is more statistical in nature, but I would just mention that. So there could be two types of trials we know. One would be superiority trials where the drug or the vaccine beats placebo, and the other could be non-inferiority trials where you compare your drug or vaccine with something that is already have been approved. Now, for superiority trials, currently the guideline says the requirement for approval is that the point estimate for the treatment difference, right, with rates of people, let's say, who have COVID in the vaccine arm against the placebo arm, that has to be at least 50%. And the lower bound of the 95% confidence interval has to clear at least 30%. And for secondary endpoint, a lower bound which is positive is acceptable. It doesn't have to be as strict as the primary endpoint. And for non-inferiority trials, the lower bound has to be greater than, I don't know if you can read it, minus 10%. So it could be a little less. It doesn't even have to be 0%. It can be a little worse, but not more than by more than 10%. Now, obviously, the non-inferiority trial thing is a moot point currently because there is nothing approved in terms of vaccine. There are, I believe, 3 vaccines that are in phase 3 right now. But once one of them gets approved, and then if somebody starts another trial with a new agent, they probably would have to go against that vaccine rather than placebo. But right now, all trials are placebo controlled. Now, surrogate endpoints, it says, may be acceptable. But again, the issue is, validation and things like that, or whether it's reasonably likely. That's something that would require discussion with the agency. Now, that's vaccines. Now, let's look at drugs and biologics. So for vaccines, we said the disease or the viral infection, right, could be a primary endpoint. For drugs and biologics, that is not exactly the same. So it says for phase 2 trials, a virologic measure may be acceptable as a primary endpoint, but that's for phase 2. But for phase 3 confirmatory trials, a virologic endpoint can be assessed as a secondary endpoint, but not as a primary endpoint. Okay, because the virologic response has not been established as a surrogate endpoint. Now, so some of the endpoints that the guidance talks about, and we know from what we see from public domain, so the endpoints are different based on the status of the patient. If the patient is in severe or critical condition, then some of the endpoints, these are the examples, okay, there are more there. Proportion of patients who are alive and free of respiratory failure. So let's say we combine the two, and that's why I call it a composite endpoint. Time to sustain recovery could be another endpoint. All-cause mortality could be another endpoint, right? And clinical status using an ordinal scale where you mix different things. You bring different single endpoints into one, again, as a composite endpoint that could be of interest. And right now, actually, the all-cause mortality and the ordinal scale, these are the two that are mostly used, as we see in the literature right now. And as mentioned, the all-cause mortality is also part of the ordinal scale. Mortality is part of that always. And for patients who have mild symptoms, outpatient treatment trials, their endpoints could be proportion of people who are hospitalized or time to sustain clinical recovery. So, these would be applicable endpoints. So we have a lot of choice here. Now, the choice of the endpoint depends, as I mentioned, population studied, for example, right? Whether these are mild population or severely ill people. It also depends on the mechanism of action. You know, is it treating symptoms? Is it treating, respiratory respiratory problems? So it depends on how the drug works, right? And so you'd have to obviously go with the mechanism of action that you think the drug works in, and what the influence of a particular endpoint would be. An important other thing would be the baseline disease severity. Whether this is the same as the population in some ways, but it also depends on age, comorbidities, etc. And this again, I kind of repeated the endpoints here, and there are probably one or two more in this slide. And these endpoints can be used as a single endpoint or as a composite endpoint. But the last bullet, if you look at that, this is in quotation marks because this is taken from the guideline itself. It says analysis of all cause mortality will be important regardless of the selected primary endpoint. So mortality is very, very important to the agency. Now, let's go to the other considerations to put a context to the endpoints. Now here, the guidelines currently says the trials must be placebo controlled. As we mentioned, that there is nothing approved yet. So all trials for now must be placebo controlled. The primary efficacy analysis should be based on the intention to treat population. So basically, they should be treated as randomized. So if there are dropouts, if there are people given the wrong treatment, people who are supposed to receive placebo receives the active treatment, by mistake, they will stay with the planned treatment, right? So that's what it means. As I said, that the time here is measured, and it's measured in days, okay, not weeks. So the way we'd have to make sure we are collecting the data that's reflected, because number of days would be very, very important, because a lot of these decisions are based on number of days. Okay, number of days in the hospital, number of days somebody could die, etc. The prognostic baseline covariates should be pre-specified in the primary efficacy analysis. So the primary efficacy analysis, when you analyze the endpoint, you have to look at the covariates, and obviously you'd have to plan for, how we are going to use the covariates, whether we'll stratify the trial, whether we would handle that analytically in the analysis itself, in the models, etc. Right? So you have to pay attention to that. And so all these would have to be pre-specified in the protocol, of course, how we are going to handle the covariate adjustment. And then if we have mixture of patients, let's say some mild, some moderate, some severe, etc., then the subgroup analysis, the interaction between treatment and the population, etc., would be very important. And this is not different in COVID trial. This is always the case. But here we know that the baseline plays a huge role, because the disease state, the impact of these baselines on the disease right now is pretty well established. We know that people who have COVID-19, the impact of the disease on these populations, or even the impact of the virus on these populations, is not uniform. So in terms of analytical strategies of these endpoints, so right now, most of the endpoints can be analyzed as a binary outcome. For example, success, failure, proportion of people who are dead, or who have respiratory symptoms or who do not, etc. Right? Time to event analysis, we can use a proportional hazard model, or these are statistical terms to do a regular time to event analysis, time to death, etc., etc. And then we can have the composites like ordinal outcome analysis, where we put a lot of these individual pieces, individual endpoints into one endpoint. Okay, and a lot of people have done that. There are various approaches to handle that. This is more complicated, of course, because some of the outcomes could be binary, some are continuous, some are so there could be a mix, and it's difficult. So number of days in hospital, if you mix it up with mortality, things like that, it could get fairly complex. There are some statistical ways to handle it. I'll talk about one or two of these just to give you a flavor of how this should be done. But as I said, these are all complicated, and you have to pay a lot of attention if you are using a composite ordinal outcome while designing the study, while calculating sample size, power. These are not trivial. I mean, a lot of them would require simulation studies, etc. We ourselves have done some. They're very, very interesting from a, I'd say, from a statistician's point of view. But again, these are not trivial. Whereas the binary outcome or the time to event analysis is pretty straightforward. Now, whatever you do, as I mentioned before, you have to make sure mortality is always captured, right? So, and mortality shows up in all three. It could, right? If you look at mortality as a binary outcome, that's one thing, or time to death could be another, or it could be part of the ordinal outcome analysis where mortality is the worst condition, right? So let me take you to a couple of these, composite ordinal outcome analysis, what they mean, right? So, so for example, I mentioned multi-state analysis. For example, in a severe case, the person may not be non-ventilated and then goes to ventilation. The person can come back or can be discharged directly, or could go to death, right, with or without ventilation. So there are different paths the person can go from one stage to the other. This is called the multi-state uh, uh, framework of this. And there is a way to, there are a couple of ways to analyze these data. And for milder case, you can see the transitions, that the boxes are different, and the way they're modeled are, there are transition probabilities that would have to be estimated, etc. And then there are some strong assumptions in terms of, to see, because these models itself are so complicated, and there are some simplistic assumptions like, like Markov assumption or something like that, that would have to be used to make it more, being able to handle that case. There is one interesting approach we did look at was the rank-based approach. One of the advantages of rank-based approach is you can mix up different kinds of endpoints because you can always rank them. So for example, mortality we know is worse than ventilation, worse than hospital stay. And suppose we calculate, we take data on mortality, we look at time to death, and we can rank all patients with their time to death, right? Or they are censored, and then we can rank them between the two arms. And then, so first we rank mortalities. Let's say time to death. All the times are done first. So mortality is ranked first. And then we look at mechanical ventilation. We rank them second. But all ventilations would rank below mortality. And then hospital stay would rank below that. And then when we look at, compare the ranks between the two treatments, and there are rank-based tests, like Wilcoxon rank sum, etc. But those are statistical terms. We can compare them pretty easily. Okay, so this is an interesting, but again, the design of the study is not trivial. It would require simulation because some of these endpoints are correlated among themselves, right? People who are ventilated would tend to, die probably with higher probability than people who are not ventilated, etc., etc. So there is a little bit of complication here. So some of the other considerations would be, these are true for any study really. But again, it would be important to keep these in mind when somebody is designing a COVID-19 study. So as I talked before, multiplicity is a big concern. If you have more than one primary endpoint, and if your success depends on at least one, then you have to address multiplicity. You have multiple secondary endpoints and you want to place some of them in your label, you have to deal with multiplicity. So that's something we have to do in any trial, including COVID. Missing data would be a big concern, and this is the usual FDA concern. What happens if data are missing? And I always suggest that we have to be conservative in our analysis when the data is missing. So suppose you don't know somebody discontinued, you don't know the status of them, whether they are dead or alive, you'd have to probably assume that they're dead, okay, when you do the analysis. And then there would be sensitivity analysis around that, whether if they are in the placebo arm, you may think they're alive. If they're on the treatment arm, you might think they're dead, and you can move these number of dates like that. And there are sensitivity analysis like tipping point analysis and all that we'd have to deal with. Sample size power calculation, as I said, if these are single endpoints, I think it's fairly straightforward. Powering the study, knowing what the size of the study would be, but with composite endpoints, it could get very tricky. And obviously, the other challenge would be to have the data that you need for powering a study, because you have to have some idea about how your drug works. If you're using a continuous endpoint, you need a variability estimate, etc., etc. So that could be a bit tricky. And the last thing I put here, I'll talk about it in the next slide, define estimates. Okay, so this is a newer concept, relatively new, that has come through ICH E9 addendum. And so basically, what an estimate is, so basically, whenever you talk about an endpoint, you have to talk about an endpoint in the context, right? And that context has three other parts. The population, so for example, intention to treat, right? FD already said that that's the population you have to consider. The summary measure, right? So what are we looking at? Are we looking at a proportion of something? Are we looking at median time to survival? Okay, whatever it is, we have to put that measure in the endpoint itself when we talk about the analysis, the hypothesis test, etc. And then there are intercurrent events. What happens when different things are happening to the patient? How do you handle that? That has to be also made clear. Okay, so this is an interesting concept, and all protocols should have a sense of the estimate. If not the protocol, at least in the statistical analysis plan, this must be there. So in summary, I'll summarize now. So the first bullet says basically follow the general framework on endpoints. Okay, so how do you choose an endpoint? Whether there are multiplicity issues, etc., missing data, right? All that you have to think about when you choose an endpoint, because when you choose an endpoint, you have to think what happens if somebody is lost to follow up. Okay, how do we deal with it, right? And given the current situation, sometimes it could be challenging. Because the person, they could miss visits, etc., depending on the endpoint. If somebody would miss a visit, what would you do? There could be a lot of missing data, right? So there are things to think about. Right now in COVID trials, right? So as I said, not just COVID trials, any trial, only the clinical outcomes are of interest, right? Because they are directly related to the patient. Now, unless there is a surrogate endpoint, right? Of course, you can get approval through a surrogate endpoint. But at this moment, there is no surrogate endpoint for COVID-19, right? So, and if somebody can think of, as I said before, a reasonable predictor, that the agency might allow that. But again, that depends on the mechanism of action, and there would have to be a discussion with the agency to convince them that yes, this looks like a reasonable endpoint that can be quote unquote used as a surrogate for now, but with a commitment that clinical outcome benefit would be proven later or the surrogacy would be proven later at some point. Virologic endpoint, as I said, can be used as a primary endpoint in a vaccine trial, but not a phase 3 confirmatory trial with drugs or biologics. Earlier trials can use it, of course, like proof of concept studies, etc. Right now, all COVID-19 trials should be placebo controlled, unless there is some other reason that, for blinding purposes, you want to use a small dose of something, depends on that. And I said for now, because once one or two drugs come out approved, then this whole scenario would change. Then we'll get into the non-inferiority trials because it would be unethical to use placebo at that point. So that's something also to think about, because non-inferiority trials again are not trivial, because they have their own issues that we'd have to think at some point. As mentioned, collect the covariates, right? So design the study with your CRF pages that cover the baseline covariates. Use day as unit of time. So the way you would have to design the study, that you are measuring things, on a daily basis, some of these endpoints. For vaccine trials, either the disease itself, COVID-19, or the virus infection can be used as primary endpoint, as I said. Secondary endpoints can include symptoms, hospitalization, etc. For drugs and biologics, two most commonly used endpoints right now are mortality and the composite of ordinal responses. Mortality is key because it has to be there all the time. Now, one of the things that I highlighted here is if you use mortality, the COVID trials are so short, what we see right now, that people are doing trials that go only months, a few months, right? Then I think it would be a mistake to put time to death as an endpoint. If you choose mortality, I would recommend that you look at proportion of deaths within that time frame. There are two reasons. First of all, when you choose time to death as an endpoint, then your sample size, the size of the trial, is driven by the number of events that you have. And you may not have enough number of events within the time frame of the trial. So that could be problematic because you know that you are going to fail because you haven't observed any, the sufficient number of deaths to have a meaningful analysis. So I'd say in that case, go to proportion of deaths, because whatever the number is, and you can at least do the analysis and show the benefit. The other point is that, the trials are so short. Even if you look at the median time to death, so let's say being different between placebo and the active treatment, it's going to be probably a month or 2 months, and that may not be clinically meaningful. If we postpone the death by a month or two, probably people don't care, right? So again, so that is something that's a recommendation that I would make. And ordinal and composite endpoints, if you choose that, there is a lot of work that has to be done at the design stage to make sure that we are covering all the aspects, including sample size calculation, etc. And and it's always a good idea to consult regulators if there is any doubt or any questions, or you want to convey the mechanism of action and how the reasonable surrogate endpoints could be used in your trial. That has to be communicated and we would say at the protocol stage to the agency. Okay, so that's all I have here, and if you have questions, Lisa can relay some of these questions. But if we have about 10 minutes. If I don't get to your question now, then please send me an email and I'll try to respond via email. Okay. Yeah, Lisa.
Thank you, Dr. Ghosh. Um, I guess our most common question is people would really like a copy of the slides. So would it be possible for us to send
Yes,
everybody registered with an
yes,
email slides? Okay.
yes, of course. Yeah. Mhm.
Super. So a question from David. Sorry, I don't have a last name. He's wondering if you can comment specifically on a primary endpoint of days to hospital discharge. Um, so I'm assuming he means how would that be handled, collected, things like that.
Yeah, I think days to hospital discharge have been used already, right? Remdesivir trial has had that. It's an interesting and see the thing is, I think drug development, what I showed in terms of the clinical outcome, right? That's the clinical piece. But also, as we know, drug development has also moved beyond just the clinical efficacy, right? Right now, even if we have clinical efficacy and safety, that data may or may not be enough because the cost effectiveness of drugs, things like that, that they are coming into the picture, right? So that's where the days in hospital or to hospitalization would be important because um, any amount of time the patient is in the hospital, it's going to cost. And so that's why it is an important endpoint. But it's not a clinical endpoint as such. I'm not. It's a softer endpoint that way, right? Because days in the hospital. Um, if you use that, and if you do not have, let's say you have success there, but you do not have anything else, uh, that would be kind of very soft evidence of a benefit to the patient. Because if the mortality is the same, let's say, it would be kind of troubling. Uh, but if, you know, uh, see, the thing is the regulatory, um, the whole atmosphere right now is very dynamic. So it's changing, ever changing, right? And we know that FDA has been very accepting of different ideas. Um, so but I know that after the remdesivir trial, a lot of people started putting that in. But I know for sure that for the FDA they're more going towards more clinical outcomes that we talked about. Yeah.
Thank you. A question from Abdul Malik. This one's interesting. It's more geared towards vitamins and natural supplements. And his question is, we know that supplements and many natural health products generally have multifaceted effects, and people generally already have androgynous levels, examples, vitamins. For COVID trials where the IPs are supplements or natural health products, one would expect effect sizes to be smaller compared to drug trials. As such, the type 2 error rate is increased disproportionately if the family-wise error is adjusted for. Does the FDA have any accommodations for this? Do you have any advice for this?
I'm not very familiar with supplements. Obviously, I mean, supplements are supplements and drugs are drugs, the way I look at it, right? And the evidence, I mean, I talked about vaccines and drugs in general. As I said, for supplements, I'm not sure exactly, how to even design a study with supplements. Yeah, because that mechanism of action could be, very soft or not well understood. Just because people use them doesn't necessarily mean, it's one thing to take a vitamin. It's another thing to try to treat a viral disease. Yeah, that's probably a non-answer to a valid question. Yeah.
Thank you. Thank you, Dr. Ghosh. Rashida has a question about a surrogate endpoint. The comment is on using cytokine storm concentrations of baseline levels at the release from the hospital as a surrogate endpoint. Have you seen anything with cytokine storms being?
Cytokine storms, I have seen some discussions. I don't recall exactly now. But again, I mean, if that could be a valid, surrogate endpoint. But again, or not surrogate endpoint, but reasonable endpoint, let's say. But I think probably I would advise to have some feedback from the agency on that. Yeah.
And that was the final question that was submitted so far, unless
Okay.
if anyone has any last minute question, if they want to submit it in the chat. Or otherwise, you can email Kalyan. His email is on the slide being presented now. And thank you, everybody, for attending. And we will work on getting the slides sent to all emails used for registering.
Okay. Thank you all for attending. Okay. And please reach out to me if you have any questions about the presentation or anything else for that matter. Okay.
Thank you, and a lot of thank yous in the chat. I don't know if you can see them, Kalyan.
No, I can see the chat window actually. So yeah, unfortunately, I cannot see the chat window. So that's why the questions have to be relayed to me.
Great.
But okay, Nathan, thank you for listening. Okay.